My name is Brian Lee, and I'm a postdoc with the Sirota Lab at UCSF. My talk today is titled Transcriptomics-based Drug Repositioning Pipeline Identifies Therapeutic Candidates for COVID-19. COVID-19 is the disease that's caused by the SARS-CoV-2 virus, and as of June 2020, there have been over 10 million cases worldwide according to the World Health Organization, with over 500,000 deaths. Only a few effective treatments have been discovered thus far, including remdesivir, which received an emergency use authorization from the U.S. Food and Drug Administration, and dexamethasone, which is a steroid with promising preliminary results from a UK study showing improved survival rates among severely ill COVID-19 patients. Drug development has a long lead time, while the nature of the current pandemic calls for urgency, which motivates us to use drug repurposing to identify new therapeutics from already existing drugs. In my talk today, I will propose using a computational drug repositioning pipeline employing a pattern matching strategy to identify new uses for existing drugs using transcriptomic data. Given the differential gene expression for an abnormal condition, in this case COVID-19, and the differential gene expression for a set of drugs, we can use those genes as a proxy to find conditions in drugs with opposite differential expressions, with the hypothesis that if the gene expression in COVID-19 is the opposite of that of a gene expression for a drug, then that drug could be therapeutic. We've previously applied this pipeline in a number of different contexts, including Crohn's disease, inflammatory bowel syndrome, various cancers, and preterm birth. In order to apply our pipeline, we first needed to aggregate differential gene expression signatures for COVID-19. We found 3 different published datasets that examined either cells infected with the SARS-CoV-2 virus or from patients diagnosed with COVID-19. Our first dataset from Blanco-Mello et al. Examined human adenocarcinomic alveolar basal epithelial cells infected with the SARS-CoV-2 virus. The authors found 120 differentially expressed genes, 100 upregulated and 20 downregulated. From these genes, they identified some enriched pathways, including cellular response to virus infection and humoral immune response, specifically with regards to chemokines and cytokines. The second signature we examined was from Lammers et al., looking at organoids that were grown from gut tissue and then infected with the SARS-CoV-2 virus. We applied a log2 fold change greater than 2 cutoff in order to examine only the driving genes of the signature, leaving behind 69 upregulated genes and 65 downregulated genes. Lastly, our third signature was from Zhang et al., who examined bronchoalveolar lavage fluid samples from 2 COVID-19 patients and 3 controls. After applying a log2 fold change greater than 4 cutoff, we identified 1,200 upregulated genes and approximately 300 downregulated genes. Using these 3 signatures, we searched for commonly enriched pathways from the 50 GSEA hallmark gene sets. In this figure, each column is a signature. From left to right, we have the alveolar signature, the organoid signature, and the lavage fluid signature. And each row is a pathway. A red square indicates an upregulated pathway for that signature, and a blue square indicates a downregulated pathway for that signature. Some of the more commonly upregulated pathways included interferon response, E2F targets, and oxidative phosphorylation, while some of the more common downregulated pathways included adipogenesis, which is the process by which fat cells differentiate and has been studied in relation to obesity and diabetes, myogenesis, and the mTORC1 signaling pathway. On the drug profile side, we use the connectivity map database, which contains differential gene expression from testing drugs in cultured human cells. They measured approximately 22,000 genes with data available on just over 1,300 drugs. In order to calculate the reversal scores, we first make a comparison between each disease signature and each drug profile. The reversal score itself is a non-parametric rank-based method based on the Kolmogorov-Smirnov test statistic. Essentially, if we have a disease signature here where blue is downregulated genes and red is upregulated genes, and we compare it to each of the drug profiles, similar pairs will generate positive scores, and opposite pairs will generate negative scores. As we iterate through each disease-drug pair, we obtain a distribution of scores with the positive red scores indicating similarity between the disease signature and the drug profile, and negative green scores indicating opposite gene expression, which is what we are looking for. Furthermore, we test for significance by comparing to a distribution of random scores, and after adjusting for multiple comparisons, we found 102 total unique drug hits across the 3 signatures. We can visualize the top 15 drug hits for each signature in a heat map of the ranked gene expression values. First, on the left here are the top hits from the alveolar basal epithelial cell line signature. The disease signatures in the left-hand column with the downregulated genes in blue and the upregulated genes in red. These genes were used to calculate the reversal score against the drug profiles to the right. And while we don't expect to see any perfect reversals of the disease signature, we can see an overall pattern of reversal with the more downregulated genes in the disease signature being matched with the more upregulated genes in the drug profiles. Next, in the middle, we see the top 15 hits from the organoid signature, and on the right are the top 15 hits from the lavage fluid signature. Some of our hits were also identified by other analyses as potentially being therapeutic. Haloperidol and valproic acid were found in an analysis of host factors for SARS-CoV-2, while both colchicine and sirolimus were found in network analysis papers. The U.S. clinical trials website also shows that some of our hits are currently in clinical trials, including colchicine, niclosamide, and sirolimus. We wanted to examine the common hits from our pipeline, or hits which reversed at least 2 of the 3 signatures. This heat map here shows the 25 genes which reversed at least 2 signatures, and column-wise from left to right are the hits for the alveolar signature, the organoid signature, and the lavage fluid signature, with brighter green squares indicating more reversed scores for that profile. At least 6 of these hits have either been identified by another study or are in clinical trial, as I noted on the previous slide, with 4 drugs reversing all 3 signatures. We also used target data from Drug Bank to look at known drugs from these targets. We constructed this drug target network visualization with the drugs in pink, the targets that are targeted by only one drug highlighted in blue, and the targets targeted by multiple drugs highlighted in yellow. This cluster of drugs at the top especially has multiple common targets. And we hope to use this information as we move forward with the validation process in order to identify potential mechanisms of action. And we also wonder if one of the drugs in this top cluster, for example, is effective, could the rest of the drugs also have therapeutic value? There were 4 drugs which reversed all 3 of the input COVID-19 or SARS-CoV-2 signatures, and these were becampicillin, clofazimine, haloperidol, and valproic acid. A little bit more information about each of these drugs individually. Becampicillin has been used to treat bacterial infections. Clofazimine is an antimycobacterial which has been used to treat leprosy. Haloperidol or Haldol is a psychiatric medication used to treat schizophrenia. Haloperidol can bind to the sigma-1 and sigma-2 receptors and was found to decrease viral growth in SARS-CoV-2 infected Vero E6 cell lines by Gordon et al. Lastly, valproic acid is a drug used to treat epilepsy and bipolar disorder and has been implicated in the inhibition of HDAC2. This drug was also tested by Gordon et al., where they did not observe decrease of viral growth, but it was suggested to test valproic acid at a higher dosage. So in summary, we aggregated 3 differential gene expression signatures for SARS-CoV-2 or COVID-19 from published datasets. We identified 25 common hits which reversed at least 2 of the 3 signatures, and 4 consensus hits which reversed all 3 signatures. Moreover, we have plans for validating some of our identified drugs in Vero E6 cultures infected with the SARS-CoV-2 virus, and we are also in the process of applying our computational pipeline to patient data. Thank you so much for listening, and I would like to acknowledge some members of the Sroda Lab, especially who were a tremendous help on this project, including Adit, Gaia, Kat, Dan, and Tamiko. Thank you again.